Hi everyone, welcome. I hope everybody hears me well. So for those that don't know me, my name is Ana Stoian, and today it is my pleasure to introduce AMAcademy, a platform from Animalia dedicated to sharing, learning, and connecting. AMAcademy has been created with a simple but powerful goal: to bring together knowledge from the lab, the field, and the public health. Because we believe that the real progress happens when these three worlds come together as in one health. Since last year, Animalia has been building its own research department, and in that time, we've been able to establish robust, repeatable challenge models for PRRS, Glassadella parasuis, and post-weaning diarrhea in piglets. But beyond that, we continue to explore other pathogens and diseases, generating the tools and knowledge needed to support the development and testing of vaccines and other pharmaceutical products. So make sure you stay tuned for the upcoming chapters of AMAcademy. We expect to have around one episode per month. And if you have any questions regarding our projects, if you're more interested, just do not hesitate to contact us directly. Today, we are thrilled to welcome Ana Stoian, a specialist in molecular biology and cell culture techniques for the study of animal and human viruses, but she has a strong focus on PRRS research. Ana's scientific journey began at the University of Bucharest, where she earned her degree in biochemistry, followed by a master's in biochemistry and molecular biology. She then pursued her PhD in virology at the Kansas State University and continued sharpening her research expertise as a postdoctoral fellow at the University of California. She's currently a Sarah Hunter Assistant Professor at the University of Lleida, where she works hand in hand with Lorenzo Fraile and Romy Pena. And she has also a very, very close collaborative history with us at Animalia. She played a key role in the characterization and development of the PRRS challenge model based on a highly virulent strain belonging to the Rosalia lineage. But nevertheless, her work doesn't stop here. She's also deeply involved in uncovering how pigs' genetics influence susceptibility to PRRS infection and the host response, because genetics could be a crucial control measure for PRRS. So in this episode, Ana will guide us through some of the most exciting recent discoveries in PRRS research, discuss their relevance for both the lab and the field-level herd management, and explore how this knowledge can shape the future for animal health. So Ana, welcome. How are you doing?
Hi. First, can you hear me well? Is there
Yes,
some
we can.
echo, or we're okay? I think
We're fine.
Well, thank you for the very kind introduction and very thorough introduction, Ariadna. To be honest, I wouldn't be here today without the support and guidance of all the professors that have been working with me, mentoring me all throughout this time. A very short career behind me. I don't know if we'll be able to cover everything that is known, but we'll try to cover as much as possible. And I'm looking forward to our conversation. And thank you for having me here today.
Oh, thank you. Thank you very much, Ana. But before we get started, I would like to let you know that at the end of the session, we will have some time to answer your questions. So to ask a question, just click on the hand button next to the microphone below. You will see there's a hand. If you click the button, it will turn purple. You can click it at any time and just leave it like that, because you will be transferred and we will give you permission when the question time is appropriate. Okay, so just also make sure that you have permission on your microphone and if you also want your camera. So let's start with the questions, Ana. Ana, can you tell us what first drew you to study viruses and what led you to focus specifically on PRRS and cell cultures?
Okay, so it's been an interesting journey, to say the least. What first drew me to viruses was, of course, their ability to take over the host cells and cause complex diseases. So, since my early training, I became particularly interested in how viruses interact with the host cells at molecular level. During my bachelor's degree, I was just monitoring and detecting PRRS strains on the Romanian territory, but then I moved on during my PhD, and I focused more on the virus host interaction, specifically on PRRS, but I also worked with other animal viruses. Later on during my postdoc, I worked with human viruses this time, but with vaccinia in particular, but again, focusing on virus host interaction at molecular level using cell culture. So it was right there with me from the beginning and continued with me along the way, no matter what virus I focused on. But although I worked with different animal and human viruses, PRRS became a natural focus because it sits at that intersection between the molecular virology and the real world impact. As we all know, it's a virus with enormous economic and animal welfare consequences, but at the same time, it poses some really interesting biological questions. It's been around for so many years, and we still haven't found a solution for it. When it comes to cell culture, this came into picture because, well, I think it's where we can really dissect these mechanisms behind viral host interaction, and not only in a controlled way. And if we want to understand why PRRS behaves the way it does in the animal, in the pig, then we first have to understand how it behaves in the cell.
Yeah, you're completely right. Thank you. So, as many of you know, the rise of the highly virulent PRRS strains has led to a very devastating impact in the Spanish pig sector. Ana, yourself, as part of the University of Lleida and in collaboration with Animalia, we have been working to characterize and understand this new highly pathogenic variant named Rosalia. So, why does characterizing a strain matter so much? What kinds of insights can it give us, and who stands to benefit from that knowledge?
Okay, so characterizing a strain is essential when we talk about PRRS because not all PRRS strains behave in a similar manner, as we have seen along the years. And differences at the genetic level can translate into major differences when it comes to virulence, transmission, tissue tropism, but also very importantly, vaccine responses. So I think getting information about how every strain behaves in an animal and having this information in an experimental setting is very important, and it can benefit multiple stakeholders. On one hand, we have the researcher who can refine their experimental models depending on the results, and also learn how to ask different questions and guide the research towards different solutions that fit our real life scenarios. On the other hand, vaccine developers can assess protection gaps, and they can also assess if it's needed a reformulation in their vaccines. And finally, I think veterinarians and producers can also benefit from this because they can set realistic expectations about disease spread and control.
Right, thank you. So from everything we've studied so far about Rosalia, what has surprised you the most? For me, if I can start, one of the most shocking findings came from the reproductive model. What we normally consider very low exposure levels, Rosalia still had a powerful impact on farrowing performance. I found that incredibly surprising and at the same time a bit alarming, right? Not even for farmers, but also for vets. As you can see here in this graph, we tested different concentrations of virus, and even at the very low level, 10 to the power of 2, most of the piglets, over 50% of the fetuses born were autolytic, and this was very surprising for me. What about you?
No, I totally agree with you. What surprised me the most was how efficient Rosalia is at causing disease, even at these very low exposure doses. So, from a virological perspective, that suggests that we're dealing with a virus with very high infectivity and replication fitness. So, maybe a faster replication, higher yield of genome copies, and makes a better use of the host resources. This reproductive model was particularly striking. The fact that such low doses still had a significant impact on farrowing performance tells us that the margin of error in the field is very small.
It is.
I guess it highlights how difficult it is to control highly virulent strains once they enter into a naive or a partially immune population. And with all these Rosalia studies that we did, not only with this one in particular, I think also what is striking is the variability between studies that we talked a bit about. And I think that relates to the genetic background in the end. And maybe we'll talk a bit more after that, or maybe there'll be questions about this. But I think these 2 points were the most striking for me.
Yeah, I understand. So from our latest findings on the efficacy of current commercial vaccines, because we did not only develop the challenge model, but we also tested some commercial vaccines in our project. So this study suggested that PRRS vaccination tends to be more effective against the respiratory form of the disease compared to the reproductive one. From a lab perspective, what factors do you think could explain these outcomes observed both in control animal studies and also very likely in the field?
Yes, it's true that from all the studies that we carried out, and I think we carried out quite a few, right?
Yeah.
3 or 4 vaccine studies, and then 3 studies in SOS. So we had quite a few studies so far.
Yeah, we did.
I think that commercial PRRS vaccines appear to be more effective against the respiratory form of the disease because, in this case, probably a partial immune response is sufficient to reduce lung pathology. So, in other words, non neutralizing levels of antibodies or low levels of neutralizing antibodies combined with interferon gamma producing T cells can reduce the viral load in the porcine alveolar macrophages, shorten the viremia, and attenuate lung lesions and clinical signs. I think that on the other hand, when we talk about protection against the reproductive failure, so the reproductive form of the disease, protection requires a rapid and probably near complete control of the maternal viremia and an effective immune response at the maternal fetal interface. And probably these are conditions that are inconsistently observed when we talked about the currently available commercial vaccines.
Right. Thank you. Now, let's get deep into your main area of expertise, the PRRS cell cultures. How do you think your international experience shaped your knowledge on PRRS and cell cultures?
Okay. So, as I kind of said or mentioned in the beginning, I have been working with different viruses, not just PRRS, and I guess the common ground for all of these was the molecular virology. So the molecular approach and then the cell culture. When I worked in different countries, I got exposed to very different approaches to PRRS research, and not only, but because your question relates to PRRS, I'll just focus on that. So in the US, there was a strong focus on working with the PRRS virus 2 strains. I only worked with a few PRRS 1 strains, and that was during my PhD, where the main focus was the development of an in vitro system that would allow me to study virus-host interaction and having the ultimate goal of identifying very small localized modifications that I could do in the virus receptor CD163 in order to render pigs resistant to PRRS2. Of course, it would be likely interesting to get this in PRRS1, but the main focus was PRRS2. In Spain, on the other hand, the emphasis has been on PRRS1 strains, especially Rosalia. and trying to incorporate field relevance and linking lab to herd outcomes, as well as genetic breeding as a different a different tool to control PRRS. So I think when I moved to Spain is where my perspective changed a bit too, because I was up to that point. So in my master's, PhD, and my first postdoc in California, I was very focused on the molecular part, the lab focused part, which is very important. But then here in Spain, I am more aware of the real life situations, and I'm trying to mix these two. So Lorenzo was very helpful in this aspect because he knows a bit of the both worlds. And we've been working on finding a common ground on how I can also tackle these real life situations with in vitro approaches and cell culture. So I think this combination helped me appreciate the strengths and the limitation of cell culture and in vitro systems in general, and to always ask how the in vitro observations relate to what we see in animals and how they can tackle these real life situations.
Yeah, you're right. I think it's very, very important to know both worlds so you can come up with the real problem, right? Find a solution for the real problem. So now talking about the cell cultures, what do you think are the main limitations of the current in vitro systems used for study PRRS?
Okay, so I guess the main limitation is that no current in vitro system fully reproduces the complexity of the pig, right?
Yeah.
PRRS primarily targets macrophages. These macrophages are highly specialized, they're heterogeneous, and influenced by their tissue environment. So, for those of you here with us today who have worked with macrophages, you know very well how hard it is to work with them. So for those of you who do not know, I've been using them a lot for African swine fever experiments because we're using the Georgia strain that only replicates in these macrophages. Nowadays, there are also studies showing immortalized macrophages can also work. But I've been also using them a bit for PRRS. So the problem with those is how I said, they're heterogeneous. So what I mean by that, we have also a batch-to-batch variation. So we have to acquire these cells from young piglets because they're the most susceptible animals. But on top of that, even getting from those animals, that doesn't ensure that we're going to get the same level of susceptibility to PRRS. So all these batches have to be tested and validated and then selected only the most susceptible ones so they can give us reproducibility over time. So every time you come up with a new batch, you have to ensure that the replication is at a very similar level to the one before so you can compare your data. Cell lines, so to kind of summarize, cell lines on the other hand are convenient, they're reproducible, right? So but they lack many of the immune and the signaling pathways that are present in these primary cells. And on the other hand, the primary cells like PAMs are biological relevant, but they have all these other issues. So I think we always need to interpret in vitro data with caution and always try to follow up with in vivo validation.
Yeah, you're right. Because how do you distinguish between cell-related issues and virus-related issues when you work in the lab?
So that is a very common challenge. The first step is always to assess the cell health independently of infection. So we have to look at morphology, confluency, viability of the cells, and it would be good to also take into account the baseline gene expression, especially for CD163, which is the main receptor for this virus. If cells are stressed, they're overconfluent or improperly differentiated, the virus can behave very differently. On the virus side, issues like passage history, freeze-thaw cycles, improper handling, or adaptation to specific cell line can strongly affect the replication. So, and this is only without taking into account the human error and the variability that we can see people handling these cells. And I think the controls are key here in order to be able to troubleshoot and figure out what is happening when something goes sideways. So always use mock infected or uninfected cells, reference strains, and parallel infection that can help you pinpoint where the problem originates. And again, if you're using primary cells, you'll always have to make sure that you compare them with the previous batch that you had.
Right. So choosing the correct cell type is crucial to achieve a successful PRRS culture, right? Using different cell lines can turn, in the end, one experiment to one side or another. So how does PRRS behave differently in PAMS versus immortalized cell lines? Which cell line would you recommend using for PRRS cultures and why?
Okay. PRRS behaves differently in PAMS, is true, compared to the established cell lines. As I said, the PAMs reflect the natural target cell, and they support biologically relevant entry mechanisms and immune responses. But as I already mentioned, they're very variable. And they're difficult to maintain and work with. So again, if you have worked with these cells, you know that they detach easily. So you have to be really careful how you handle them. And then again, we have these issues with the batch-to-batch variation. The Mark 145 cells, on the other hand, which are the more known cells for PRRS, are easier to use and are highly reproducible, especially for PRRS2. But they select for adapted variants that may differ from the field strains. So I think ideally both systems should be used in parallel. So cell lines for standardization and throughput, and PAMs for biological relevance. And here it also depends on the type of study, like what are you looking for? For example, if you're just trying to grow some PRRS strains that grow very well in mark cells, you can do that. You can titer, you can quantify the amount of virus, maybe also use them in neutralizing antibody assays depending on the strain if it replicates. But you wouldn't use Mark 145 cells if you want to look at the innate immune response, for example. So for that, you will use macrophages, for example. So it depends.
Yeah, I see. So you had, you were very lucky to be able to work with the two different species of PRRS, especially in the USA. You mentioned that you focused on PRRS type 2, and PRRS type 2 is relatively easy to isolate in and adapt to Mark 145 cell line. And what about PRRS type 1 and, of course, Rosalia?
Okay, so that's correct. PRRS2 adapts relatively easy to Mark 145 cells, which has been extremely useful, but PRRS1 remains much more challenging. For PRRS1, primary alveolar macrophages are still the safest way to go. However, in recent years, we saw that immortalized macrophage-derived cell lines, such as the PAMKNU cells or even the ZMAC cells, have become very promising alternatives. So the PAMKNU cells are immortalized macrophages, exactly how I said. There are different ways to immortalize these cells, and in particular, the PAMKNU cells have been immortalized by using the human telomerase method. In addition, there are also engineered cell lines nowadays. You have mark cells that are having the CD1C3 overexpressed that also help with the virus replication. Although these cells retain more macrophage-like characteristics and key receptors involved in PRRS entry, which makes them more permissive to PRRS1 than classical epithelial cells, and they also offer improved reproducibility compared to primary macrophages and can support virus isolation and propagation for certain strains. They are not yet a universal solution. So permissiveness can still be strain dependent, and prolonged passaging may lead to viral adaptation. I think that for this reason, careful control of passage number and genetic monitoring of the virus remains essential. So I think that overall, I see this immortalized macrophage system as a very important tool stepping forward. And combined with primary macrophages and genetic validation, they can significantly improve our ability to study PRRS1 in vitro while maintaining biological relevance. And this figure is actually from our paper that you mentioned in the beginning of this webinar. As we can see here, we had the Rosalia strain replicating in 3 cell types. We had it replicating in mark cells, PAMK cells, and the primary macrophages. And you can see the PRRS virus 1 strain Rosalia does not replicate in mark cells. And then it has different profiles in both primary macrophages and the immortalized macrophages. But what was important for us was to show that at 3 days after infection, when we usually do the readout in all our assays, the results in the 2 cell lines are comparable.
Right. Thank you. So, as we mentioned, your contributions to the PRRS challenge and efficacy studies we've carried out have been crucial. What do you think these experiments brought new to the research, the PRRS research?
So I think these studies generated controlled comparative data using a highly virulent, but also very importantly, a relevant field strain, which represents a key contribution to PRRS research. A lot of the studies are carried out on old PRRS strains that are not as relevant nowadays. It helps us gain more information and knowledge about PRRS, but studying the new ones is even more important, especially when we have highly virulent strains. So by standardizing challenge conditions, these studies allowed a clear assessment of viral virulence and comparative vaccine performance that is difficult to achieve under field conditions. Importantly, evaluating commercially available vaccines against the highly virulent strains such as the Rosalia provided, in my opinion, also valuable insight into the level of and consistency of protection that can realistically be expected in the field. From a personal perspective, this brought with it other things. I also developed a neutralizing antibody assay with the help of these PAMK cells, which can later on open different opportunities for preventive medicine studies if we can combine the neutralizing antibody assay with an ELISpot, so looking also at cellular immunity, not also at humoral immunity. So it opened different pathways, this type of not only the generation of knowledge per se, but also opened some room for different studies along the way.
I see. Well, you already mentioned some lab work that has been very useful in this big project for PRRS. So now looking ahead, how do you think the cell cultures can support more and advance the future research for other studies?
So I think that looking ahead, PRRS cell culture will remain essential for answering these targeted and mechanistic questions as cell systems become more refined. So as I said, especially these macrophage-based immortalized cell lines or engineered models where we overexpress CD1, CD3 in a certain cell line, they will allow us to link viral genetics with biological behavior, such as replication efficiency, cell tropism, and immune modulation, which is very important for PRRS. This will help us better understand why certain strains are more virulent and harder to control. And it will support the rational design of vaccines, challenge models, and control strategies before moving to animal studies. So I think these two can work hand in hand. We will always have to rely on in vitro and cell culture information before moving to animals.
Yeah, of course. So PRRS is a very, very complex virus, and studying it in live animals can be very challenging, expensive, and even sometimes very limited in scope. So that's where cell cultures and in vitro work really come in. What are the particular challenges in translating what we see in vitro to what happens with the animals?
I think the biggest challenge is oversimplification, to put it in one word.
Okay.
Because in vitro systems isolate specific processes, right? While the animal integrates immune responses, then we have the physiology, we have the genetics, and the environment. So it's good to be able to separate these processes to be able to study each one of them, but then you don't get the big picture. So cell cultures are excellent for hypothesis generation, but validation in animals is always essential before trying to draw any conclusions for field applications.
Right. And what kinds of questions about PRRS biology can cell cultures help answer that would be difficult to study in the field? As you mentioned, when we work with animals, the immunity really comes in, like the interactions with the immune cells and stuff. So let's put it the other way around. What can we know better with cell cultures and in vitro work?
Okay, so it's true. Cell cultures allow us to study virus entry, so receptor usage, replication kinetics. We can do virus isolation and propagation. So we have different applications of these cell lines. The primary cell lines for innate immune responses or for replication of those viruses that are hard to replicate in the most common cell lines. And for this, we have different established cell lines as well. We can also look at the pathogenesis and infection mechanisms. We can do vaccine development. So for vaccine development, also the cell culture is an important component. Then the diagnostics, because being able to do virus isolation on field samples is also very helpful in this kind of cases. Antiviral testing is also carried out in cell culture. And then on top of that, it also allows us to test targeted interventions such as receptor editing in a controlled setting. So for example, as I said in the beginning, my PhD focused on the PRRS virus receptor CD163. And the goal was to identify small mutations that wouldn't change the overall conformation of the receptor, that would render cells and ultimately pigs resistant to PRRS. And for this, I relied solely on a cell culture system. So this is just to give you an example. On the left side, you have different constructs of the CD163 receptor that I did in vitro. And then I used HEK cells, so human epithelial kidney cells that are normally resistant to PRRS virus replication, to express these different receptors. The transfected cells were then infected with PRRS, and then I would monitor the level of infection. So the goal was to see less infection, to be able to identify the mutation that actually renders the receptor inactive for virus replication. And I used a similar approach also for vaccinia, but this is just an example for PRRS and the impact the cell culture can have.
Right. And do you think in vitro cell cultures can predict how the virus will respond when moving to animal trials? Can we know that?
This is a very good question, and one of the editing strategies that I did during my PhD. So after that first diagram that you showed, we identified domain 5 of the receptor as the most important part in the PRRS virus replication, and then I moved on and I made even smaller mutations within that domain in order to block the infection. And by doing all these experiments, I identified a completely different region that was important for PRRS virus 2 replication. And a smaller part of that, of 12 amino acids, was also able to block infection. So we have a patent for that. And actually, my PhD professor, after I finished my PhD, went along and made these edited pigs. So the pigs are lacking that part of a PST2 region, the part of the receptor that I identified in vitro. And he challenged those pigs with a PRRS2 strain, with NVSL, which is a very common strain in the US. And as you can see here, the animals were negative for virus replication and also for antibodies. So I think in this situation, it was a successful translation from in vitro to in vivo results, but the cell culture cannot always predict what's going to happen when we've moved to animal trials. So I think the best approach is iterative. So first conduct in vitro observations that will inform animal studies, and then animal data will refine again the in vitro models.
Right, thank you. So, well, despite everything, all these years of research, PRRS continues to be a very major challenge in the swine industry, and we might not learn everything for many, many years still. But in your opinion, where do you think we should focus next in research? What are the areas that we should explore in more depth?
So one major challenge is the viral diversity and the evolution of PRRS. It's hard to keep up with this ever-expanding diversity of the virus. I think another challenge is understanding host genetics and immune responses in a way that they can be translated into control strategies. So I believe that research should focus on integrated approaches that combine virology, immunology, genetics, and field data. And as you already know, this is something that our group is trying to do. So we are trying different things. Looking into genetics and see how genetics can affect resilience or susceptibility. We also are looking into antivirals and also a project that looks at developing a vaccine capable of eliciting broadly neutralizing antibodies. So we're trying a bit of everything through collaborations, not necessarily our main framework, but I think only by integrating all these tools we can tackle this and find the right answer. And not to mention that I guess the genetic editing is also knocking on our door. So we'll see where that goes, because so far it's been pushing forward in the US. I don't know if you are aware of that as well. But here in Europe, although it's going to take longer to come, I think we have to be prepared and probably get involved and get the animals tested before they actually come here and know if this is actually a solution or not. So we have different lines of research right now. Everyone is trying to tackle this.
So you think there's a probability that we will have genetically modified pigs resistant to PRRS in the end in Europe?
I think this is happening. Yes, this will happen. If you would have asked me a few years ago, I would have said no, that is not possible. But I think now it's the wheels are spinning, you know. So they're gonna be here sooner rather than later. So we have to be prepared, and then just be sure that these animals are actually resistant to Rosalia strain or whatever strain is going to be circulating at that point. And then see what other options we have. As I was mentioning, these edited animals that are on the way, they're edited for domain 5, but my professor showed through my in vitro data that also another part of the receptor is important and can make pigs negative. So the question is there an alternative model there for the interaction between the receptor and the strain? And how is that gonna look long term, right? Are these animals gonna stay resistant long term, or the virus is gonna learn how to adapt and find a different entry point through the same receptor?
Yeah, I mean, this is a possibility, right? We need to be careful with this virus. Ultimately, the goal, as you mentioned, is to achieve a better sanitary status, especially now here in Spain that we have Rosalia. So how do you think the in vitro research can better bridge this gap to the field applications in the end?
You're absolutely right. In vitro research has value if it ultimately improves what happens on farms. The way you can best bridge the gap is by being purpose-driven and closely connected to field questions, which is something that I was telling you that I'm trying to do more and more since I moved here to Spain. In vitro systems allow us to rapidly test hypotheses that would be difficult, expensive, or ethically challenging to explore in animals. And then this information can be used to prioritize strategies before moving to animal trials. I think the most important part is that in vitro findings become most meaningful when they are validated and interpreted alongside field and in vitro data. In vivo data, sorry. So when lab results are generated using relevant field isolates, low passage viruses, appropriate cell types, that's when they can inform decisions on disease control.
Right. And well, there's another topic that I think it's very interesting right now because it might also be the future if the genetically modified pigs don't arrive earlier than we expect. So how do you think the autogenous, the PRRS autogenous vaccines can be a reliable control measure in the future?
I think they can be a valuable tool in specific contexts, especially during outbreaks with highly divergent strains. As we know, these autogenous vaccines rely on killed strains. And this type of vaccines are not as efficient at conferring protection as the modified live ones. So I think that they could be used as part of an integrated control approach, but not as a standalone or a long-term strategy. These systems require also time, money, and as we know, sometimes we have a very narrow window of action when these vaccines would actually be most effective. So I think I would focus on genetic breeding rather than autogenous vaccines, but I might be biased in this aspect.
Okay, so you mentioned a few of them, but what are the main challenges when preparing PRRS autogenous vaccines?
So one of the main challenges in preparing these autogenous vaccines is the virus isolation first, and the stability. Not all field strains grow efficiently in cell culture, and some require multiple passages. So when you multiply passage these viruses, you increase the risk of selecting cell-adapted variants that no longer fully represent the field strain. Another major challenge is preserving biological relevance while meeting production and safety requirements. So the virus must be sufficiently amplified, inactivated in a controlled manner, and proven free of contaminants, all without altering the key antigenic property of this virus. So time is, as I said, it's a critical factor as well. The autogenous vaccines are often needed during active outbreaks, but isolation, characterization, safety testing, and formulation take time. So sometimes longer than the epidemiological window where the vaccine would have the greatest impact.
Yeah, you're right. You mentioned something about this, but how do you ensure that the virus we took the samples from to produce the autogenous vaccine still represents, or not the autogenous vaccine, but maybe for challenge models when developing them or characterizing a strain? So how do you ensure that this virus still represents the one circulating?
So I think that's a very important point because cell culture itself can unintentionally change the virus. To minimize this, the first principle is to work as close as possible to the field isolate. And this is something that we're trying to do in all our animal experiments. In practice, that means using low passage viruses, limiting the number of amplification steps, and whenever possible, starting from clinical material directly from the herd, such as serum samples. We also carefully choose the cell system. So, for example, we've been using the primary alveolar macrophages or the immortalized macrophages, the PAMKNU cells, rather than MARC145 cells when biological relevance is critical. How it would be in these challenge experiments. So in parallel, we also constantly monitor the virus genetically. We conduct whole genome sequencing before and after cell culture, and this will allow us to detect any adaptations or mutations that may arise during the isolation and propagation of the virus.
Well, Ana, thank you for your time. If there is one key message that you would like the listeners to take from this discussion today, what would it be?
I think my main message would be that cell culture systems and in vitro approaches in general are powerful tools, but they are most effective when used thoughtfully and in combination with animal studies and field data. More importantly, not necessarily related to cell culture, is that in order to tackle this virus, we need to come together and try and present all the information that we have, report everything that we see, which sometimes can be tricky, especially with negative data and especially with data that we don't understand. But I think it's very important with these studies, with the cell culture and in vitro data, that we're fully open to share with others our doubts, our inconsistency, to be able to better understand what is happening and to finally figure out a solution for this virus.
Yeah, I see. Well, there's a long way ahead of us
Yes.
in PRRS research. So thank you very much, Ana. But before we wrap up, we would love to hear from the audience if they have any questions. That would be the time to ask them. But if some questions remain unanswered, feel free to contact Ana. We can share her email or contact us if you have some questions to us. Any questions? Okay, well, it seems that we don't have any questions so far. That would be the time.
I'm happy that makes me believe that we were very thorough and we answered or we covered everything that was to cover. But again, if there are any questions, please feel free to reach out to either of us and we'll get back to you.
Okay, so, well, if there are no questions, just a reminder that you need to click the hand next to the microphone. But if there are no questions, well, thank you very much, Ana, for joining us today and sharing your expertise on PRRS research and, of course, the cell culture models. It has been a very interesting conversation, really, and I hope our listeners enjoyed it as much as we had. Ana, thank you for also being part of this first episode of AMAcademy. It has been very inspiring, and I also want to mention that, well, as I said, if anyone is more interested about this PRRS challenge model or the studies that we have been carrying out, feel free to reach out. And looking ahead, yeah, go ahead, please.
No, I just wanted to say thank you too for having me here. I hope it went well for the first episode. I felt the pressure because I want you to continue having more and more episodes. So thank you for choosing me for this first episode, and I really enjoyed our conversation, and I hope the audience felt the same way.
Yeah, well, thank you. And now looking ahead, our next session will be on February the 25th, and Maria Montoya will be joining us. She's an expert in influenza and immunology, and we hope that it will also be as interesting as your speech. Thank you very much.
That will definitely be very interesting. Maria knows a lot of things as well. I'll make sure I'll also subscribe for that one, and I'll be there.
Yeah, thank you. Well, thank you, everyone.
Bye.
Bye.